Hello and welcome to the Microbiology Lab. I'm Dr. Gareth Price, Project Lead for Galaxy Australia, and I'll be giving you a quick introduction to this lab. This interface of Galaxy Australia is part of an amazing effort by the international microbiology community to collate community-backed tools, workflows, and training material on microbiological data analysis into one easy location. If you're curious to find out more about how it was developed, then please read our preprint linked here on the lab. The Microbiology Lab is part of a larger Galaxy Australia ecosystem, allowing you to access the same tools, resources, and infrastructure that you would on Galaxy Australia. You can get to all the functions of Galaxy Australia through the hexagon icon at the top of your screen dashboard, where you get back to Galaxy Australia or navigate to other labs that already exist: Genome Lab, Proteomics Lab, and Single Cell Lab, and now the Microbiology Lab. The lab is divided into sections. We have a Getting Started section for those of you who may be new to your microbiology analysis journey. Shows you through bringing in data into Galaxy, highlighting some of the common tools, interactive tools for visualizing, manipulating your data, and learning pathways, which I'm going to talk about in a minute. Broadly, the sections are separated into the analysis of a microbe, the microbial isolates section, and below we'll discover the next section. Here, you have a very nice overview of all the tools and journeys that you might take to analyze your isolate. These are found individually as tools in the tools menu with a drop down to run that tool straight away in Galaxy Australia. Interactive tools, data sets that you might need to annotate or further enrich your data, workflows, which will be imported from their source of truth at the workflow hub. The very important Galaxy Training Network tutorials, which can pop up immediately inside your Galaxy instance to follow along and learn a journey, or can be opened in a new tab, depending on your preference for following along. So we've been in the microbiological isolate section. The other section that I mentioned is the microbiome section that has a very similar format, a very similar and useful overview of all the analysis you might want to do in your journey with metabarcoding, metagenomics, transcriptomics, or metaproteomics. Again, individual tools, workflows, and tutorials can be discovered supporting this kind of analysis. Workflows are further featured in our community workflow section, where workflows have been built, curated, and sourced globally. They're peer-reviewed, they follow the FAIR principles, they link to GTN material. It's a very rich section to allow you to get the best out of your data in an efficient manner through utilization of workflows. And our last sections are when you might want to get some more support. That support could be through direct interaction with the microbiology community globally, which you can do via chat rooms. You can access the Galaxy Australia team through local support we have that will raise a ticket with our team. There's FAQs. And finally, there's an opportunity to participate back in the growth of the global microbiology community. So you can do that by joining the community and chatting to them. You can find out on the Galaxy Training Network how communities are built and organized if that's of interest. Please do at any point give us feedback on the lab and your experiences with it. We use that feedback. We welcome it to continue to provide this resource to you. With that, thank you for your attention and enjoy analyzing your data on the Galaxy Australia Microbiology Lab.